in a cell cycle, consider interphase is present and the division phase. So interphase is a time gap between two successive mitotic divisions. So that means the interphase, when the cell is undergoing mitosis, after completion of one mitosis, the cell will prepare itself for the next division. That stage where the cell is preparing itself for the next division is called as the interphase, which is further divided into G1, S and G2 phases. Whereas division phase is further divided into karyokinesis and cytokinesis. So likewise, the cell cycle can be divided into for these stages. After cytokinesis, the cell will enter into next cell cycle. So the next cell cycle once again will start with the interphase. Earlier cell cycle. The earlier cell cycle will have been stopped with the cytokinesis. So likewise, this is a first cell cycle, second and the next and the next. Likewise, the changes will be taking place. So in this process, the cell cycle, out of the entire changes which are taking place, the cells in the cell cycle, interphase is considered to be the longest phase, which will take more time. Generally, it is not compulsory that all animals or all plants should have the same time period for interphase. So here for human beings, it is 24 hours. Out of the 24 hours, G1 will account for 10 hours, S phase accounts for 9 hours, G2 phase accounts for 4 hours and mitotic phase or the division phase accounts for 1 hour. Likewise, the total cell cycle is operated for the 24 hours. Out of this, the 23 hours are for interphase. Let us take the example of yeast. In the yeast, the entire process will be completed within 20 minutes. Whereas in some bacteria, the entire process of cell cycle will be completed in about 20 minutes, 18 to 20 minutes. Likewise, the time taken for cell cycle is different for different types of living organisms. Like in the human beings, the time taken for cell cycle is 24 hours. In yeast, it is 90 minutes. In bacteria, it is 20 minutes. In some of them, it may be extended for days or sometimes weeks or years together also, depending upon the metabolic activities which are performed by the organism and also the environmental conditions prevailing surrounding that living organism. Here in the living organisms, there are two types of divisions are there. One is called indirect divisions and other is called as a direct division. In the indirect division, the spindle fiber formation is present. Which is by either mitosis or mitotic division or meiosis or meiotic division is taking place. While in the lower organisms like in bacteria and in some protozoans like in paramecium, where the nucleus will divide directly without the formation of spindle apparatus, that is called as a direct type of division. So that process of division is called as amitosis. The type of division is called as amitosis. Let's discuss about the amitosis which is taking place in the bacteria. In bacteria, the amitosis will occur by a process called as binary fission, where as it is a unicellular organism, the constriction will appear in the cytoplasm and in the cell wall, that is moving towards the center in the centripetal manner, so leading to the formation of two daughter cells. So in this process, consider this is a bacteria with cell wall and cytoplasmic content. Here in this one, the nucleus will be present. The nucleus first it will elongate. That will elongate first, and it will be divided in, it will divide into two daughter nuclei within the bacterial cell. But at the same time, a small constriction will start in the protoplasmic content on both the sides. Particularly this will occur in the place of mesosomes. So these are the daughter nuclei that have been formed. And at the same time, a constriction will start on both the sides. So this constriction will gradually move towards the center. So it will move in a centripetal manner, that is from periphery to the center, and gradually it will divide the cytoplasm into two equal halves. And already the nucleus have been divided, so we can say that two daughter protoplasms have been formed. At the same time, a constriction will start in the cell wall, and that constriction which is formed in the cell wall will also move in the centripetal manner, that will divide the bacteria into two daughter bacteria like this. And the so formed two daughter bacteria are structurally and morphologically similar to each of them, and they are not only similar to each of them but also similar to the parent cell, and they will increase in its size and converted into the independent bacteria organisms. So this is a way how the nucleus is first elongated and divided, later the cytoplasmic contents also divided to form into two daughter or distributed between the two daughter organisms. So this type of division where there is no spindle formation, directly the nucleus is divided, so we call it as a direct type of division or amitosis, which is considered to be the primitive type of division.